So we talked about the different types of antibiotics and their targets of action, as well as some of the mechanisms that bacteria use to resist being killed by antibiotics. And now we are going to talk about a key feature in the clinical microbiology laboratory, and that is antibiotic susceptibility testing. Now, we have to do antibiotic susceptibility testing in the clinical microbiology laboratory because this is how a clinician knows how to treat a specific infection. It avoids using unnecessary medications that might not actually work against that organism. It optimizes the therapy, but the thing about susceptibility testing is it has to be highly standardized. So you wouldn't want to say that a specific organism was susceptible to an antibiotic and come to find out that the test wasn't accurate and it really wasn't susceptible, and then you're treating someone with an antibiotic that won't actually work. So standardization is key with susceptibility testing. Now, for standardization, there is the Clinical and Laboratory Standards Institute, or CLSI, which establishes all of the normal values for the susceptibility testing. So the three main purposes we already said optimize for standardization are optimizing your bacterial growth conditions. If you use more bacteria than you're supposed to for a specific assay for susceptibility, you could get different results. You could show that the organism is actually more resistant to an antibiotic than it actually is. So you have to optimize the bacterial growth conditions. You have to optimize all the conditions that are going to maintain the antimicrobial agent's integrity and its activity. So the antimicrobial agents have to be stored at very specific temperatures. Usually they're stored in a refrigerator, and you wouldn't want to store them at room temperature because they might lose their activity. And then you would do a test on a patient specimen and it might show that it's susceptible, but really the antimicrobial agent you're using has just lost all of its activity. You also want to maintain reproducibility and consistency. So no matter who does the susceptibility test, no matter what instrument it's done on, no matter what laboratory it's done in, you should get the same results. There are many variables that are going to affect antimicrobial susceptibility tests. So the size of the inoculum, we already said, if you use too much bacteria, you can actually have more organisms that might be able to resist in higher numbers, when in reality that organism isn't really resistant. The rate of bacterial growth. So you have to be aware of how quickly an organism is going to grow so that you don't allow it to overgrow. And also, some of our organisms that have very different growth rates, you might have a modified method to perform the susceptibility tests. You always have to keep in mind the incubation periods and follow those very stringently. The media you use, so commonly if you're using an agar-based susceptibility tests like a Kirby Bauer test, which aren't really done in the clinical laboratory anymore, but you would use a Mueller-Hinton based agar. However, there are some fastidious organisms that we've talked about where those organisms need a blood based media. They wouldn't grow up on those types of agars. So you might have to use specialized media for some of the susceptibility testing. The stability of the antimicrobial agent, so you never want to use an antimicrobial agent or disc that was expired or that had been sitting out at room temperature for too long a period of time. And the incubation temperature. So a lot of susceptibility tests are performed at 35 to 37 degrees, but there are different susceptibility tests for some of the fastidious organisms. So you want to really make sure you're following all of the appropriate steps for your susceptibility tests. Now, two important terms with susceptibility testing that you definitely need to know: the MIC and the MBC. The MIC means minimum or minimal inhibitory concentration. And what that is the lowest concentration of your drug or your antimicrobial agent that's going to inhibit visible growth. And the MIC, if you were to do it manually, it's done using serial broth dilutions. Now, the minimal bactericidal concentration, it's a little different. It is the dilution where you kill the bacteria. So MIC is inhibiting visible growth, not necessarily killing the bacteria. MBC is when all the bacteria are completely dead. And in order to know if a bacteria is completely dead, you would need to grow it on the appropriate agar-based media and see if you get any colonies growing. So here would be your broth and agar types of tests. So on the top, you have a susceptible organism, and on the bottom, you have a resistant organism. So all the way over to the right, you can do your agar-based susceptibility testing that we've talked about numerous times throughout the semester, where you have a little circular disc that's impregnated with a very specific concentration of an antibiotic. You make a suspension of your organism, you make a lawn of growth on your agar media, and you place the disc with your antibiotic on the media, and you tap the top of the disc ever so lightly with a sterile forceps. You then incubate that plate, and the next day you look at it and you're looking for your zone of inhibition. Now, for a broth-based test, what you're going to do is you're going to make serial dilutions of your antimicrobial agent. So all the way over on the left, you always want to have just broth alone with no agent in it. That's your control. That's your viability control. This is very important. You always want to have a tube that has absolutely no agent in it so that you see that your bacteria grew. If your bacteria doesn't grow in the zero or no antimicrobial agent at all, it means your organism that you use to inoculate is dead. So your results are not viable. You would have to repeat the test. Then you start out with 2 micrograms per mil of antibiotic, 4, 8, 16, and you could keep going. Now the MIC is the first tube that does not show visible growth. In other words, the first broth that's completely clear. There's no turbidity, meaning bacteria grew in it. That is the minimum concentration of antimicrobial agent that inhibited the growth of that particular organism. So now on the top, you could see that the MIC is 8 micrograms per mil. But on the bottom, that organism grew in every single solitary tube because the organism is resistant. The other thing to keep in mind is you have increasing concentrations of your agent. You also want to make sure you add the exact same amount of bacteria to each tube. So you'd make an appropriate suspension of your organism, and you mix it so that all of your organism is completely mixed in your broth, and then you would add the same exact amount to each tube. If you added more bacteria to one of those tubes, you might see inhibition, when in fact it's just because you had more bacteria in it, and it would take more antimicrobial agent to inhibit a higher number of organisms. So this is a concept that you really have to understand. It's not something that's easy to memorize. So I hope I've explained it in enough detail where it's easy to understand. You can also read about this in your textbook. If you're still having some trouble understanding it, just shoot a question onto the discussion board, and I could make some more diagrams or explain it in text format. So here's an actual picture of an MIC and an MBC. So you have a rack with your broth tubes, and if you look all the way over to the right, you have a broth that shows turbidity. Right next to it, it also shows turbidity. But then the next tube and all the other tubes with the serially diluted higher concentrations of antimicrobial agent, they're clear. So that third tube in would be the MIC. The third tube in from the right hand side would be the MIC. All the way over on the left is some controls. So a viability control that's turbid, and a negative control just to make sure that your broth wasn't contaminated. You could just put your broth in a tube and incubate it without inoculating any organism, and that will let you know that your broth wasn't contaminated with any other organisms that might affect your results. Now, in order to do the MBC, you take a small portion of each one of these tubes and you plate it out onto agar plates, and then you stick the agar plates in the incubator. Now, the third tube in from the right is your MIC, and let's just say for this antimicrobial agent, it's 8 micrograms per mil of a specific antimicrobial agent. If you plate that tube onto an agar plate, you likely will actually get growing bacteria, because that concentration, 8 micrograms per mil of antibiotic, is only enough antimicrobial agent to inhibit growth, but it didn't actually kill the bacteria in that tube. So you would inoculate an agar tube from each one of those tubes. You actually might find that although your MIC is 8 micrograms per mil, your MBC might not be until 32 micrograms per mil. You need much more antimicrobial agent, a higher concentration, to completely kill your bacteria than you do to inhibit. So it's very common for the MBC to be a higher concentration than the MIC. You will have some questions on your next exam about this concept, and also on your final exam. So if you don't understand the difference between MIC and MBC and why you might see colonies growing on a plate from your tube that was your MIC, then just send me an email or post a question on the discussion, because it is a very important concept for you to understand before you go into a clinical microbiology laboratory. Now, we mentioned there are the disc diffusion methods to do susceptibility testing. Now, the old-fashioned method, and if we were doing a student microbiology laboratory, so I've taught in 2 other clinical lab science programs, and in both of those I did microbiology lecture and laboratory courses, and we would do the Kirby Bauer susceptibility test in our student laboratory. So hopefully many of you, all of you that are MLTs, I'm sure, have done a Kirby Bauer, and a lot of you who've taken a microbiology course in some of the general microbiology courses, you may have done a Kirby Bauer. So Kirby Bauer, it's important to know that it's a qualitative test. It is not a quantitative method. It does allow pretty rapid testing of multiple antimicrobial agents at the same time. So again, these are our little circular discs where you have a specific concentration of an antimicrobial agent impregnated into the little paper disc. And when you drop that paper disc onto the agar, it generates a gradient of the antimicrobial agent into the agar. And you measure the zone of inhibition. So the organism is going to be able to grow to a certain point. But in a certain point within the agar, it's going to hit a concentration of the antimicrobial agent that's going to inhibit its growth. And you will see that zone of inhibition around it. So this is a picture on the left of the Kirby Bauer test, and this is the old-fashioned way to do your susceptibility testing. And this is a method that you'd use in a student laboratory. So in that picture, you could see the blue contraption, and that is a plunger on the top, and it holds numerous tubes containing your antimicrobial agent discs. So if you take that blue circular piece, you put it right over your Mueller-Hinton agar plate, and that plate already has your lawn of bacteria. You already make your suspension of your organism. You plate your lawn of bacteria on your plate. You then add your antimicrobial agent discs to your Kirby Bauer contraption. You put the whole blue apparatus right over your plate. You press the plunger, and the plunger drops one disc from each tube evenly spread out on your Mueller-Hinton agar plate. You then gently tap on each disc with a sterile forceps, and then you incubate your plate, and the next day you look at it. The picture on the right hand side of this slide is a close-up of the little tubes that are filled with your antimicrobial agent discs. Each one has an expiration date on it. You only use it if it's within its date. Once it's expired, you must throw that whole package away. You could see the little gray area on the on this picture, the close-up of the one that's outside of the packaging. That is a little easy way. You just press on that gray piece, and it pulls out one disc, and you can drop it manually on your agar plate. So again, here's your Kirby Bauer contraption with the plunger holding the various disc tubes, as well as the Mueller-Hinton agar, and it's stored in a special container. You could see that in the back. When you're not using it, you store it in this special container in the refrigerator. Now for your disc diffusion method, your Kirby Bauer. Again, you start out with a lawn of bacteria. It's really important to make a suspension of your organism. Plate a lawn of bacteria using a sterile swab all over the plate. You usually go back and forth in one direction, move the plate slightly, go back and forth in another direction, move the plate again, go back and forth in another direction, so that the entire plate is covered with your organism. You then take your Kirby Bauer plunger unit, put it right over the plate, plunge down, and it'll drop your disc, or you can drop your discs manually. What happens is, you can see in the schematic on the left, you have your disc directly in the center. You then have a smaller zone. Now it says if there's growth within that ring, so even though there's a zone of it, there might be a zone of inhibition. If the zone of inhibition is not large enough, it still means that that organism is resistant to this antibiotic because it was able to grow in too close to the disc. If growth is in between the smaller ring and the larger ring, it means that it's intermediate It's not completely susceptible, but it's also not completely resistant. It's an intermediate. The zone has to be larger than the outer ring in order to call this particular organism for this antibiotic resistant. So there's a book, the CLSI makes all of these standards of exactly what measurement is resistant, what measurement is intermediate, what measurement is susceptible. So you have to measure every single one. It's not as easy as just looking at it and saying, oh, there's a zone of inhibition. This antimicrobial agent is good for this organism because it inhibited growth. No, you actually have to measure the zone for each and every antimicrobial agent that you drop onto the disc. Now, how do you prepare your inoculum, your bacteria that you're going to spread on your plate? What you use is a 0.5 McFarland standard. So in the clinical microbiology lab, we use these McFarland standards. They are commercially prepared tubes that contain kind of a very fine sand in water. And if you shake the tube and put it up against this card that contains the black lines on it, you should make your inoculum of bacteria so that it's about the same turbidity as the standard. So the McFarland standards come in many different numbers. 0.5 McFarland is a very common inoculum standard that's used in the clinical microbiology laboratory, but there's a 0.2, 0.3, but there's different numbers of McFarland standards. Some of them are very, very light and some are very dense. So you could see that the tube on the right is the bacterial suspension, and it's ever so slightly heavier than the 0.5 McFarland standard. Once you make your 0.5 McFarland inoculum and you plate it on your disc, and then you drop your plate it on your plate, inoculate it onto your plate, spread it out, you add your discs to your plate, you incubate for the appropriate incubation period, usually it's overnight. You're then going to measure. So you can take a ruler, and here's another thing, you have to measure in millimeters. So a lot of times, one common mistake in the student laboratories is a student would measure accidentally in centimeters instead of millimeters, and the number would be wrong. So you measure in millimeters. You figure out the diameter all the way across from one edge of the zone of inhibition to the other. And then you then are going to look up in a book. There is a number for every organism, for every appropriate antimicrobial agent, and you're going to see if it's a number that's considered susceptible, intermediate, or resistant. So again, here's your zone of inhibition, and that's how you measure it, right across the center from one edge to the other edge. So this particular organism for this antimicrobial agent is 19 millimeters. There are these little calipers that are available. So way back when, in my day, when we used to do this test in an actual clinical laboratory, you use these calipers to measure. It made it very easy. You just open up the calipers from edge to edge, and it would tell you how many millimeters it is. There were even little digital ones, a little fancier than this. And you can easily measure all of your measurements on one plate, as opposed to putting a ruler up and holding it up to the light and hoping that you're measuring it properly. Again, here's an actual picture of a Kirby Bauer disc diffusion. You could see the different sized zones depending on the antimicrobial disc agent that's on the disc. Now, Kirby Bauer tests are not done in a clinical microbiology laboratory any longer. And the reason is because we have instruments like the Vitek and the Microscan that do susceptibility tests while they're doing all the biochemical tests to confirm the organism's identity and its susceptibility. But one test you will do in the clinical laboratory is an E-test. Now, the E-test is really nice. It's very easy. And what's great about the E-test is it is a disc method, but the Kirby Bauer, we said, is qualitative. You have to measure that number, and all you are identifying is if the agent is going to kill or not kill that bacteria. So you're determining if the organism is resistant, susceptible, or intermediate. That's it. That's all. It's a quantitative measurement. Is it susceptible? Is it resistant? The E-test actually combines the diffusion test with a broth MIC. So you actually get the minimum inhibitory concentration from an E-test. So what the E-test is, it's a plastic strip that contains an antimicrobial agent in a gradient of concentrations along the strip. So you perform it almost identically to diffusion. You make your inoculum of your bacteria. You spread your bacteria in a lawn on your appropriate plate. You then lay your strip in the appropriate direction on the plate and you incubate it. And the next day you come in and you look for a zone. Now, instead of being a completely circular zone, the E-test makes an elliptical zone because of the gradient of concentration along the strip. So you read this elliptical zone and it tells you an actual number. It tells you the MIC. It doesn't only say, here's a millimeter, go look in a book and see if it's resistant or susceptible. It actually gives you the minimum inhibitory concentration for that antimicrobial agent. Now, the E-test is a much more expensive test than the Kirby Bauer test, but it gives much more reliable results. So that's why the E-tests are commonly used in clinical laboratories to verify a test that might come out of the microscan that doesn't seem to be correct, or if the microscan is ever broken down, you might need to do some manual susceptibility tests. So the E-test would be the way you would go. So here's an actual picture of an E-test. So again, you spread your lawn of bacteria exactly the same way. The E-test also is commonly done for most of your agents on Mueller-Hinton agar plates. Mueller-Hinton is what is usually used for antimicrobial susceptibility tests, except for fastidious organisms. Fastidious organisms that require special media will require some modifications and require a different media. So you would want you want to lay your strip onto your lawn of bacteria. If you see the top, there's the letter E for E-test. You always want that to be on the edge of the plate, because that edge, the top of that strip, has the highest concentration of antimicrobial agent, and then in a gradient it goes down in concentration. So that's why at the very end of this strip you could see bacteria growing right up around that strip, because the concentration of antimicrobial agent at that end of the strip is too low to inhibit the growth of this particular organism. Here is a Penicillin E strip, and this is on a blood agar plate because this is a more fastidious organism that requires blood to grow, and you can see the nice elliptical zone that it makes. Now, how you read this test is the bottom number where that elliptical zone touches at the bottom of the strip. So organism is growing all the way up to about the 0.016. So somewhere around the 0.016 would be the minimal inhibitory concentration. Now for your broth dilution methods, we already said you would make serial dilutions, two-fold dilutions of your antimicrobial agent. You want to use a standardized inoculum and add the exact same amount of your bacteria to each tube, incubate it overnight, and just examine for turbidity. We already said you want your control tube that doesn't have any antibiotic agent in it so that you test for viability. You want to make sure your agent is actually able to grow at all. The lowest dilution that has no turbidity is your MIC. And here again we have a broth microdilution where you have your 0, that's your control, where it's turbid, your 0.2, your 0.4, your 0.8, and then your 1.6. In this particular picture, the 1.6 microgram per mil is the MIC. That is the concentration that you inhibited the growth. There's no turbidity. Now, if you wanted to do the MBC, you would plate a portion of each one of these tubes and incubate an agar plate. You likely would see that bacteria would grow on that 1.6 microgram per mil tube. So the first agar plate that didn't have any bacteria growing on it, not even one colony, would be the MBC, the minimum bactericidal concentration. You can do your broth dilutions in a 96 well plate, and so you're doing much smaller amounts of broth, smaller inoculum of organism, and this would be the microdilution method. We're going to now move on to the second part of this lecture.